Now we will explain the last very important character for describing a species, and this is evolution. There are very important names investigating evolution of the species. First, of course, you have mentioned Charles Robert Darwin and Alfred Russel Wallace, because they have developed the selection as the driving force for evolution. And Johann Gregor Mendel, he is the founder of genetic investigation of genes. Additionally, in the 20th century, start of beginning, then was a rethinking and the introduction of population genetics and of mathematical theory of population genetics into selection mainly, or into the change of the population driven by mutation and by selection. And these are three very important names working especially with mathematical methods such as Ronald Aylmer Fisher. He is a very important person also in statistics. He has developed the analysis of variance and many other methods. Then John Burton Haldane and Cyril Green Wright. In the middle of the 19th century, just in the time of the Second World War, three of these people developed the so-called synthetic theory of evolution. And this was Theodosius Dobzhansky, George Gaylord Simpson, he's a vertebrate specialist, and Ernst Walter Mayr. We will go to George Gaylord Simpson because he clearly shows three tendencies in evolution, and he made a clear separation between speciation events, where from a mother species two daughter species arises, adapted to a special ecological niche zone, while we can find a gradual transition of the species within a changing adaptive zone, and this is the so-called anagenesis. So you have not, or you must not divide such a anagenetic line, such a line. This a division within this line is an artificial line, because the ancestor and the following population in this line are nearly identical. Where is the exact real limit? So, but there are some limits, very sharp limits in such lines, could be possible. And George Gaylord Simpson developed the method of so-called quantum evolution. So we find an adaptive zone, a changing adaptive zone, and only peripheral populations are possible to rapidly transgress and occupy another adaptive zone. So it's more or less in geological history a punctual event. While in the former adaptive zone, the medium and the populations on the other periphery of the zone completely died out. So this is quantum evolution. And of course, we have a first species, and then we have a sharp limit to the following species. Also, they are really connected. Now, other names are Moto Kimura. He developed the neutral theory of evolution. Now the dating of the molecular clock is based on this neutral theory, but it still does not work in any organisms, and in almost only a few organisms, really good. Then we have Stephen Jay Gould and Niles Eldredge. They developed the quantum evolution concept, or another concept of evolution. It is called the so-called punctualistic evolution. We have also to introduce for population genetics the really important equilibrium, the Hardy-Weinberg Equilibrium, developed by Geoffrey Harold Hardy and Wilhelm Weinberg, independently from each other at the same time. And finally, one of the very important names where I don't have a picture, and this is Franz Bettenstedt, a German micropaleontologist working for oil industries, mainly in the Cretaceous, so in the last period of the Mesozoic, and in deep water, on deep water benthic foraminifera. And in such deep water sediments, there is a continuous transition of the sediments. Therefore, lines in microfossils can be found in these sediments. And a scholar of him, it is Beate Grabert, she detected first speciation events in such single-celled deeper living foraminifera around about 300 to 400 meter steps. And you can see a split off from one line, several split offs of different species. Now, Bettenstedt introduced the so-called population genetic mechanisms to paleontology. So he explained the population sequence due to selection or caused by selection. He explained directly and transferred it directly to the fossil form, explaining this sequence of population and called them fossil genetics. Here you can see the transform of a qualitative character, of a qualitative gene into a quantitative characters, and you can see here the homozygotes and the heterozygotes. And they are together causing a normal distribution of phenotypes. The curve, the frequency, shows us the phenotypes. So the influence of the genotypes, homozygote, heterozygotes, to the phenotypes. Here you can see the phenotypic expression of 3 genotypes, and you have to the left and to the right the homozygotes, and in the center you have the heterozygotes. Now, the summary distribution is not real and normal distribution. When you give, and I added here in this model, I added here a fourth phenotype, then you can see the summary distribution is not really, this is the punctual forms, is not really a normal distribution, but it's a platychrotic distribution. But still the homogeneity, the proof of the homogeneity is high, and you can also look for the concordance of each phenotype to the other phenotype, and they are all in good concordance, so they are all in good contact. In reality, to the right side, there are of course different frequencies of the different phenotypes, and if you will get a real normal distributed phenotypes, so at the borders you have to lower frequencies, and in the centers you have higher frequencies. Then you have the ideal fit, according to the proof of the homogeneity, of the total proof, and also between all these phenotypes. Now, this type of distribution along such a genotype gradient, we can say a gradient, is of course not this ideal, follows not this ideal distribution, but some of them are more important than the others, still keeping homogeneity, as you can see in both of these forms, and still approximating more or less a normal function. Here you have another types. So at the border of this distribution of genotypes, the phenotypes are much more abundant than on the other side. So this leads, can be selected or may lead to directed selection. Or on the right side, you can see that all both phenotypes on the borders are preferred against the genotypes in the center of the distribution. And you can see here, in this case, the grade of homogeneity is gone. So this probability of homogeneity is very low, but still there is concordance between all these forms. So still they are belonging to one species. In this case, this can lead to so-called disruptive selection by preferring both border phenotypes. Now when we add a new genotype by mutation, so we have two cases. In the one case, it is very close to the former genotypes, and in the other case, it has a more larger mutation rate, a larger jump in the mutation, so it's more far away from the genotypes. So you can see here also the new phenotype corresponds on the right side, on the left side, to all the other former phenotypes very well, except phenotype number 1. But on the right side, it only corresponds to the phenotype number 5, only corresponds with phenotype number 4, and not with the other one. So there is no mixture possible between these phenotypes, because it lost all these forms. And now, this was only the scheme. Here we have now the normal abundance in this case. We have first the skewed distribution, adding very close. So this leads to so-called gradual evolutionary lines. So these lines will see a shift against a gradual shift, a gradual change to the younger time. On the right side, they are completely separated, and this will lead to a separate species when there are additional phenotypes or genotypes added to the extreme phenotype, and they will be then separated, leading to punctual evolution. And now at least we have the hybridization of two populations in the contact zone caused on genotypes. As you can see, you have very distinct populations, or distinct distributions, but on the borders, in the one population on the right side, on the other population to the left side, there is congruence. And only on this congruence between phenotype number 4 and phenotype number 5, they overlap. And so this is the possibility that when they are coming in such a contact, they can hybridize, and in the later evolution, this new phenotype, this new hybrid phenotype can become very important in the line in the later stages. Now we have to look for the genotype composition through time, and in the first case you can see we are working with qualitative characters according to the Hardy-Weinberg law. You have the probability of one allele, and you can see when they are in constants through time, then they follow the Hardy-Weinberg law with the probability of the homozygotes, both homozygotes, and the probability of the heterozygotes. This can be, of course, extended to many alleles, not only to 2 allele stations. When we transfer this to quantitative genetics, then you will have a series of such normal distributions, but they are constant. So you can characterize it like a band going but keeping in constant through time, and in this picture it is designated by keeping the same color. Now what's going on? when the genotypes are changing, first we have in the qualitative character, you have the line. In the quantitative character, you have a band, a sequence of normal distributions characterized by band, and because of changing, you can see also the color will change. And this is very important for the following explanation of speciation events. Now we will go to the theoretical speciation model. This was used by many phylogeneticists working with so-called cladistic trees. So we have a mother species within a stable adaptive zone, but then there still suddenly arises, or more or less suddenly arises, two differentiated adaptive zones, but still keeping stable. Thus, the species within a grain zone differentiate and then becomes differentiate with two different species adapted to the different adaptive zones. Now, but this you can find not only, it is very rare. It is very rare in paleontology. Much more abundant is the split of speciation, that from one mother species, and in this case, mother species is changing because the adaptive zone changes, and on some kind another adaptive zones developed, first dependent on the first adaptive zone but having an own adaptive tendency, and thus the species split off, and as a daughter species from the mother species, and split off from the daughter species, and making a quite distinct evolutionary line. So many of the cladistic people, or phylogenetic systematics, they're saying we have a mother species and we have two daughter species. Why you call the one a mother species? Because according to the evolutionary species concept, they keep the evolutionary line. And if you will follow, then you can see at the end at the beginning of the end, if you will transfer this line into a linear equation, then the line does not change. It's the same line. But the daughter line follows quite a distinct linear equation. So we have the gradualistic speciation. We have, of course, examples, especially from micropaleontology. And here you can see the gradual evolution and the split off of a radiolarian. It's a planktonic living single-celled organism. The next form within the group of split-off speciation is the so-called punctated equilibrium. So first we had an adaptive stable zone, but then at the border a second adaptive stable zone is developing, and suddenly from the first zone this adaptive niche is occupied by species B. So you have a very fast punctual event from the random, from the border populations, from the peripheral populations, creating and make an integration into the new adaptive zone. And then the adaptive zone of the first species can be finished, and then this species will disappear. We have also in the old fossil record by Bettenstedt, we can find examples for these adaptive zones, and you can find them especially in deep drills of the Northern Sea, and in the upper part you have one continuous sequence of the drill, and you can see these are followed by different lines. So you can see such different forms. So really following the punctated equilibrium. Now we have another form of a split-off speciation, and this is the so-called quantum steady quantum speciation. You can see you have here an adaptive stable adaptive zone, but as in the model of Simpson is shown, then the population cannot adapt to the following zone, and it will suddenly shift to the next adaptive zone. Steady in case because the environment suddenly shifts, as you can see here in the planktonic foraminifera. In the Miocene, this means up to 6 or 5 million years. In the deep ocean, it is stable, and then on the border a very rapid change in the Miocene-Pliocene border, and then again you have stability up to the Pleistocene. And this is caused, of course, by a very strong temperature decrease in the Pliocene. In the next picture we have split-off speciation, and in this case the so-called gradual quantum speciation. So we have a changing adaptive zone, and this cannot be this is finished, and you have the next adaptive zone, but also changing again, a strong jump from one adaptive zone to the other zone, a punctual event, more or less punctual event. And we have the species A and species 2, two distinct species. And we have an example in the gradual quantum speciation, again from a planktonic foraminifera, but now living more or less in the first example, living in tropical conditions. So the temperature is more or less constant, and these forms lived in close to the polar position, not polar, temperate climate. And you can see in this time interval there was a strong decrease in temperature, and this is followed also by a change, by a more or less continuous change, but from the Miocene to the Pleistocene, you can see here a very strong gap, and it is also in another developed in another character, this strong gap. Then we have non-speciation, because you can find sometimes an adaptive zone which is stable, and then you have a transfer into a zone which changes. Of course, in a stable, the species keeps constant, and when the adaptive zone changes, then it starts with a transformation. And these are not distinct species, because at the border, the first population of the starting with the transformation is the same like the population before, the just population in the time span before. So you cannot make here a real gap, but you can divide them into two subspecies, the one keeping constant and the other changing. And we have here an example from the Cretaceous, from a planktonic form, again in the uppermost Cretaceous. At the time before the conditions have been stable, as is shown on the oxygen isotopes to the right side, and then at some border, then the oxygen isotopes become higher and higher. Such means a cooling event, and you can see the foraminifera react with a shift. In such case we have a stabilized selection, and then we have a directed selection. Then we have a so-called split-up speciation. This was the bifurcate speciation we have shown before, but the bifurcate speciation is only a special case of a normal, of a split-up speciation, of a so-called reticulate speciation. So we have a stable adaptive zone, and then within a time interval, changing and creating different adaptive zones, and all are occupied from different species, being adapted to this adaptive zone, very rapidly adapting to the adaptive zone. These adaptive zones have a distance, so between these adaptive zones there are gaps. But then some of these adaptive zones, they are combined again, some are lost, and again the populations can hybridize. So hybridization between all these adaptive zones is possible. And after a time interval, then until no more hybridization is possible, and then we have the development of clearly distinct species. Within this reticulate pattern, this is called reticulate speciation, within this reticulate pattern, different, highly different populations, morphologically different populations can be developed, but they have been regarded as subspecies because hybridization between the subspecies is always possible. And especially today, in the time interval today, when we have very close species, closely related species, so-called species, groups of organisms, most of them are subspecies of the group, and so you can often regard hybridization between quite distinct species, quite distinct forms. So this reticulate evolution was mainly first developed by an ostracologist, but here it really made it popular by Veron, and a specialist in corals, and he has shown the diversification and this reticulate pattern directly in the different time intervals. But also in larger foraminifera we find this pattern, because shallow bandic larger foraminifera, they have symbionts, and through the Indian Pacific Ocean there are different niches. You have to regard from the Red Sea to southern Japan, and you can find different morphogroups, quite different morphogroups. But when you go to the molecular genetics, then you can see that all belongs to one group according to ribosomal DNA analysis. So these are in reality really different subspecies of the same species, and here you can see the distribution, the wide distribution of the Operculina ammonoides throughout the complete Indian Ocean and Pacific Ocean. And you have the Operculina ammonoides discodalis restricted to quite a different habitat, and also Operculina ammonoides a novel species, and this is only found in Japan. But you can find also this reticulate speciation in the fossil record, and the first picture and the first paper on the speciation shows us this real reticulate speciation. Look from these three serial forms in red color. This is Gudrina. At the border between the lower and middle Albion, they split off the Spiroplactinata, but with a very broad variance, continuous broad variance in the lowermost part, and then afterwards there are clearly separated lines into three species following the same tendency. the last point is how we can delimit species within a time horizon by morphology. Here we have to regard a three-dimensional space. On the one side, we have to look for the frequency distribution along the morphological character. We have on the other side to look for the frequency distributions along the gradient. And within each of these normal distributed curves, or left-side distributed curves, you have the ontogenetic transition, according, in this case, to high juvenile mortality. And you can see there are clear three distinct normal curves. On the one side, on the character, on the other side, on the gradient. So we can speak really of three different species, and the overlap is not very strong. It's very weak in all these forms. And as an example, we have here the development or the distribution of three, again, larger benthic foraminifera. They keep their morphology totally constant. They are really different, but throughout their depth distribution. And as you can see, at the frequency distribution, the overlap is also very low. So this group with very closely related species in this kinship here, they really make the depth gradient, divide the depth gradient by three different species. Here we have the other problem. Here you can see that along the morphological distribution is unimodal, and also the distribution along the environmental gradient is unimodal. So in the form, this group changes from weaker gradient values with spherical forms at weaker gradient values continuously to flat forms at higher gradient values. Again, we can regard this as depth estimation, and this is quite relative to the former group, and this is the so-called Amphistegina radiata group. And as you can see here, there is a very strong transformation from very thick forms and smooth forms to flat forms with papillae, with small forms in the deeper form, and this is a unimodal distribution. So it is now very interesting to differentiate between these groups by these forms, as I have shown you. Now we have here the problem that according to the character, we have quite two distinct groups with different frequencies, but along the gradient, we have the same distribution. How we can interpret this? This could be two generations or two sexes of the same species. As you can see here, on the one side, the asexual, the sexual reproducing generation, and on the other side, the large, the agamont, the asexual reproducing generation. Now we will find a problem here, because you have here from the Amphistegina lesoni group, you can see here two forms. One is really biconvex and the other is umbiliconvex, this form. And if you look for the frequency distribution along the gradient, then you can see, they are really overlapping. So are these two generations? No, you have to look for another quite different gradient, as you can see here by the substrate, indicated by the mean rise in phi values. And then you can see that Amphistegina lesoni is found mainly on firm substrate, while the umbiliconvex form, you will find them on soft substrate, on sand. So it's only ecological differences. So these forms are clearly differentiated, but with a more or less a strong overlap. Now we come to the development which is correlated with the environment. And this is very easily, you can see here from the larval stadium with very rounded forms going continuously but decreasing, of course, in abundance, decreasing up to a state where reproduction occurs. So on the side with the frequency distribution along the character, you can see here the normal left side skewed distribution of high mortality and of high juvenile mortality, but not restricted within one ecological horizon, but along the gradient. And the other form is along the environmental gradient, you have a truncated normal distribution. So you have the highest values at the beginning, and then it is going down and decreasing. Truncated distributions. And an example is the planktonic foraminifera. They are living as juveniles very high in the water column, and during development they are sinking down in the water column. But such continuous change we can find in many other forms. Now, at the last point, we have the abrupt change in the environment during ontogeny, and we can find this in many, many organisms. So you can see you have different environments. You can see in the one environment, you have along the character a normal distribution, and in the other environment, you have also can have a normal distribution or a bimodal distinct distribution. So we can follow it, possibly interpret them that in the one environment you have a larval state, and in the other environment you have then the adults, and they can be separated in sexes, so also in morphology a differentiation in morphology is possible, but along the gradient, this part of the gradient, it is homogeneous, as I have explained before. But not only this can be done for larval states and metamorphosis and different other environments for the adults, but also for different generations, like we have in anthozoans, in corals, but not in economies, in hydrozoans, where we have a polyp stadium, a fixed polyp stadium, and a medusal stadium living in the water column. And then we have always like a picture like this. So it not necessarily must be divided into into two distinct distributions in one of both environments. Thank you for your attention.